Hey guys, so in this video I'm going to give you a little introduction on the double helix model of DNA, which is undoubtedly the most famous model of DNA out there. So let's get started. Alright, so let me just give you a quick overview. So here is my little imaginary cell. Here is the membrane, the outer membrane of my cell. And inside my cell, inside this black circle right here is my DNA, which makes this black circle the nucleus. And from 11th grade, you should be knowing that this little yellow circle right here is the nucleolus. So inside my nucleus right here, we're going to have this sort of mass of tangled up string that has no form, doesn't really have a definite pattern, it's just untangled and all over the place. And this right here is the DNA. Now DNA stands for deoxyribonucleic acid. Now let me just underline the DNA part for you. DNA. So the reason why it's called deoxyribo, or deoxyribose should be the full form, but it's shortened in the name, is because the sugar found in DNA, sugar is nothing but ribose right here, has one less, it's short of one oxygen atom, which is why it's called deoxy. And the nucleic acid part, well, we'll get to that later. So let's continue. So in order to find out the structure of DNA, it was actually Francis Crick and James Watson who, both of them, in 1953, so just about 70 or 80 years ago, found the structure of DNA, and they named it as the double helix model of DNA. And for that, I'm pretty sure they even won a Nobel Prize, so let's hope I'm not wrong. So now let's get to the structure. Now the structure of DNA is pretty simple. All you need to know really are the three basic components. And there's a pretty easy way to remember those components. All you need to know are these three letters, P, S, and B. Now let me just write these out for you. P stands for phosphate, which is the phosphate group found in DNA. S stands for sugar, which is the sugar that is found. And B stands for base, which is the nitrogenous base that is found in the DNA structure. So let me just go a little bit in depth for you. So in this video, this is going to be my phosphate throughout this group, throughout this video, so that's going to be pretty easy. And now the sugar is actually called as pentose sugar. And the reason why it's called pentose, if you notice the prefix here, P-E-N-T, pent, it actually means 5. Pent means 5. So from that we can deduct that the sugars that are found in DNA actually have 5 carbons. So I have this pentagon right here, pentagon, pentose, there you go. And notice how there's 5 carbons, one at each vertex. Now this is the exact same sugar that is found in DNA. And now if we move on to the base that is found in DNA, all of the bases are nitrogenous, which is basically just a fancy word which means that all the bases that are found in DNA have nitrogen in them. And there's also 2 kinds of bases, which we're going to get to in the next slide. Alright, so the 2 kinds of bases that are found in DNA are the purines and the pyrimidines. Now you're probably thinking, what are these letters, A, T, G, C? What do they stand for? Well, let me just write them out for you. A stands for adenine, T stands for thymine. G stands for guanine. And C stands for cytosine. So if you haven't already noticed, the A, the T, the G, and C are basically the first letters of the full names of these bases. G for G, C for C, T for T, and A for A. Now, what are these dashes in between them? What are they? Well, if you know a little bit of chemistry, you just may be able to understand me, because they are nothing but hydrogen bonds. And hydrogen bonds are some of the weakest bonds that are found in nature, and they're present between two bases. Now, something which is very important to remember is that adenine always pairs with thymine, and guanine always pairs with cytosine. And there's also 2 hydrogen bases between A and T, and 3 hydrogen bonds, between G and C. And that is a rule, actually, called as Chargaff's rule. So let me just write that in blue for you. Chargaff, that is the name of the person who discovered it. So Chargaff's rule states that adenine always pairs with thymine and guanine always pairs with cytosine. Now, another thing that I want to add is that there's a really easy way to remember this. A, all, T, tigers, C, can, and G, growl. So all tigers can growl. That's a pretty easy way to remember it. And another way in which you can remember the pyrimidines is that notice how there's a Y right here, and there's a Y in thymine and a Y in cytosine. So you can easily remember the pyrimidines. And if you remember these, then you sure can remember these. Let's move on. All right, now we're going ahead pretty quickly. Now, if you notice here, I think you can pretty easily distinguish the 3 components of DNA. The first thing you may immediately notice are A, T, G, C, with the 2 hydrogen bonds between A and T, and the 3 hydrogen bonds between G and C. So these are nothing but the nitrogenous bases right here. Now this, if you remember, these are the pentose sugars with 5 carbons, 1 at each vertex. And these green little circles right here are the phosphate groups. Now you must be thinking, what are these dashes that are connecting the sugar to the base and the sugar to the phosphate? How are they connected? Well, let me show you. The bond between a pentose sugar and a base, a nitrogenous base, is actually called an N-glycosidic bond, which is a pretty fancy word, I admit. And now the N right here stands for nitrogenous, because remember, I already told you guys, the bases are nitrogenous. And the nitrogen found inside these bases is what bonds with the pentose sugar. Hence, this actually stands for nitrogen, so N-glycosidic bond. Now, what is the bond present between that links these 2 sugars? What is that bond? You see this dash here, and this dash here, and a phosphate group within them. And that bond is, let me just write it up here for you. That bond is nothing but a phosphodiester bond. Phosphodiester bond. Ooh, that's a really long word as well. But a phosphodiester bond is any bond that links 2 sugars, this sugar right here and this sugar right here, with a phosphate. So that is a phosphodiester bond. And now there you have it. This is the basic structure of DNA. And this